Hello everyone, it's a pleasure to be here with you today, and I will talk about one of the experiments we conducted in our laboratory. Today I will talk about the effects of a probiotic Bacillus strains on ileal digestibility of crude protein, starch, energy, and fat, and total dietary fiber digestibility of energy in diets fed to weanling pigs. Direct-fed microbials, sometimes also called probiotics, are among the additives that are often included in diets for weanling pigs. Probiotics are believed to be able to colonize in the intestinal tract of pigs, which allow them to secrete specific enzymes that may improve energy and nutrient digestibility, and thus possibly increase feed conversion. Bacillus strains are between the microorganisms used for this purpose. Once fed by the pig, Bacillus spores can survive to a low pH and pepsin. Then, in the small intestine, where the pH is optimal to them, the spores can germinate, grow, and produce enzymes. Some studies conducted in our laboratory have found that Bacillus sp increased the fecal short fatty acid and gain-to-feed duration in a trial that we conducted for growth performance in weanling pigs. And also, another experiment conducted to test the Bacillus amyloliquefaces increased the apparent ileal digestibility of amino acids and gross energy. A third experiment conducted tested the Bacillus subtilis. We also found that this Bacillus increased the digestibility energy and metabolizable energy in growing pigs. However, a new technology has been implemented to obtain Bacillus thuringiensis, but information about the effect of this probiotic in nutrient digestibility are limited. And therefore, one experiment was conducted to test the hypothesis that Bacillus thuringiensis in diets for weanling pigs may increase the apparent ileal digestibility and apparent total tract digestibility of energy and nutrients. Moving on the material method for this experiment, we had used two experimental diets. First diet was a control diet formulated based on corn, soybean meal, and DDGS. A second diet was formulated the same as a control diet, with exception that we also added the Bacillus thuringiensis on this diet. The experimental design for this experiment was we used 16 cannulated barrels with an initial body weight of 10.7 kilograms, allotted with 2 diets and 80 replicate pigs, and those pigs were housed in a metabolism crates. The 7 first days were considered adaptation period to the diets, whereas the day 8 and day 12 was considered for collection days of feces and urines, and on day 13 and 14 we collect ileal digesta. So the samples of feces and urines, we calculate the apparent total tract digestibility of energy and nutrients, and the ileal digesta sample we use it to calculate apparent ileal digestibility of energy and nutrients. Moving on to the results. First of all, let me set up of the graphs. The light orange is the result of diet without probiotic, and dark orange is the diet with probiotic. So if you take a look on this graph, we will present the result of apparent total tract digestibility of gross energy, fat, and total dietary fiber. The apparent of gross energy, fat, and total dietary fiber was not affected by the inclusion of probiotic. And the same was true for concentration of digestible energy and metabolizable energy, which was not affected by the inclusion of probiotic to the diets. However, if you take a look on this graph that represents the results of apparent ileal digestibility, we will observe that if probiotic was added to the diet, there was an improvement of digestibility of dry matter and also digestibility of gross energy. The increased apparent ileal digestibility of gross energy for diet with Bacillus thuringiensis is likely a result of increased apparent ileal digestibility of dry matter, but may be also a result of tendency of starch digestibility increase if probiotic is included to the diet. Although we observed improvement of ileal digestibility of energy, this did not translate into an increased total tract digestibility of energy. It therefore appears that use of Bacillus thuringiensis may shift the digestibility of energy from the hindgut to the small intestine. This may increase the utilization of energy because energy in the form of glucose absorbed from the small intestine is utilized with a greater efficiency than energy in the form of fatty acid absorbed from the hindgut. This assumption is supported by the fact that ileal digestibility of energy tended to increase as well, demonstrate that Bacillus thuringiensis may secrete alpha amylase, which hydrolyzes starch and release more sugars that can be absorbed in the small intestine. Based on the results represented in this trial, we can conclude that Bacillus thuringiensis does not affect the apparent total tract digestibility of energy and nutrients. Also, Bacillus thuringiensis improves the apparent ileal digestibility of dry matter, energy, and also had a tendency to increase starch. Therefore, Bacillus thuringiensis has a potential to improve utilization of energy by weanling pigs. With this presentation, I want to make acknowledge for Rubinon for financial support. And also, if you want to know more about our research, you can go in our website nutrition.nsi.illinois.edu. Thank you.